is the Associate Professor of Maastricht University, Wilfred Germeraad. And today we are going to practice a special way of lecturing. It will be held via webinar. And first of all, since it is a new experience, I would like you to give us a feedback. Can you hear me properly? Are there any problems? Unfortunately, you can use now only the chat because we're trying to prevent any type of sound loops or technical problems. But if you hear me well, please type something in the chat. Okay, good to see. So as I already said, the topic of today's lecture is the immune system. And if you have any questions during the presentation, we suggest to type them in the chat. And then either during the lecture, if Professor Germeraad will notice your question and can answer, and he will think that the answer is your question is reliable, he'll comment on it, or we'll devote, let's say, some time at the end of the lecture where you can raise the hand. It is the menu on top of your screen with the person holding a hand like this. You can press this button and we will know that you have a question, and I will change your role from the participant to the presenter, and you can activate your microphone and camera and to ask your question personally. So I think that's the way we are going to work today. Once more, I welcome everybody who came, and I give the word to Professor Germeraad. So nice to see you, Wilfred. Please.
Thank you, Max, for this nice introduction. Well, welcome everybody. It's a strange way of giving a lecture, not being able to see you, not being able to communicate directly with you. On the other hand, I think these technologies these days allow us to continue education on a daily basis, but even from a long distance. I guess you're all at home. I'm at home. I'm in the Netherlands, in Maastricht, close to Maastricht, in a small village is where I live. And usually on a daily basis, I work at the university and we use the immune system to develop new therapies for cancer. I've been an immunologist for over 35 years now, and I think it is a beautiful system that we have, and it's a very important system that we have, and that's what I will try to explain to you today in a basic way, because the immune system is rather complicated, and I have understood that this is a real introduction into immunology. So I would like to keep it at a superficial level, but try to cover many things. And then later on, we can always think about more going into depth, depending on the needs. So the overview, I think it's important because the immune system we really have in battling danger. And the current danger, well, it's very clear to everyone, is the COVID-19 pandemic. It's all over the world and it's caused by a virus, the SARS coronavirus 2. And this is what we call the danger from the outside. So pathogens that will enter our body and make us sick. And if we cannot fight it, we will die. And even if we fight it, we see that especially with this type of viruses, still some people are dying of it because of an overreaction of the immune system. The danger here is that the virus spreads through particles, droplets, and by sneezing, coughing, and this is a nice picture of a sneeze where you have hundreds of thousands of droplets that may contain virus particles that travel to over 160 kilometers an hour from one person to another. And that also is the reason why we have to stay at home. I will come back to the coronavirus at the end of the lecture, because I think you first need to understand a couple of other things before we can talk about this. There is also danger from within, and that is what we basically call inflammation. So the question always is, what is inflammation and why should we care? Well, from a clinical point of view, inflammation is a response to injury, and that may be from the inside or from the outside. And you will see 5 symptoms that come about with inflammation. There is heat generation, redness, swelling, pain, and a loss of function. So as a physician, it's easy to see these kind of things, and you have to think about inflammation as an underlying cause. But to be honest, inflammation is in many diseases, if not all. So diseases like cancer, there is an inflammatory tissue, but cardiovascular problems, atherosclerosis, has an underlying cause of the immune system going wrong. But it's basically everywhere. I can mention all these categories and individual diseases, but that will take too long. So I think it's good to summarize that inflammation is at the root of most diseases. So it's very important to understand what it is, how it works, and maybe how you can use it to treat. So yes, the immune system is crucial. How do we divide it and how can we use it? Well, there is a so-called innate immune system. This is a system that we are born with. And these are the physical barriers that will protect us against primary danger from the outside. So pathogens cannot enter our skin. We have a process called phagocytosis. That's kind of eating of pathogens, complement, and other mediators of inflammation. And I will tell you in a minute what this is and how that works. Next to the innate immune system, we have an acquired or adaptive immune system. This is a more complicated system. It has evolved later in evolution, and especially with the higher vertebrates, but even in smaller animals, you can find it. And its main function is to discriminate self versus non-self. And the immune system should not attack ourselves, but something that is dangerous and comes from the outside. And it's very specific in its recognition of antigens. And antigens are part of the danger, part of bacteria, parts of viruses, part of changed self molecules. And clonal selection means that there is an expansion of certain cells in our body that recognize this non-self. And the very good thing about this acquired immune system is that it has memory. And I will come to that as well in the end. What is memory and why do we need it? We have been using the immune system very successfully in making treatments available to patients. And the most obvious one that I guess you all know is vaccines. Vaccines against viral diseases in general. There is a small number of vaccines that is there for bacterial invaders. And I already can say these vaccines are based on the principle of memory. Another useful system, as we understand the immune system, is organ transplantation. By transplanting organs from a healthy person to a deceased person, the patient can recover and get a fully functional lung, kidney, liver, or bone marrow. But it has to go across barriers. And what these barriers are, I also will address in this lecture later on. And monoclonal antibodies, a product of the acquired immune system we use in treatment of rheumatoid arthritis or in many cancers. That we can target very specifically the immune system to the cancer and treat it. So if I say there is a successful part of the immune system, we also have, of course, inborn errors or acquired errors. And one of those, or to start with, is autoimmunity. And autoimmunity develops when the self versus non-self discrimination is not strong enough. Actually, the immune system turns against self and destroys organs of our body. You also may have an acquired immunodeficiency caused by viruses, like the HIV virus causing AIDS. Or you may have genetically immunodeficiencies, and these can be very, very severe. The ADA, adenosine deaminase deficiency, is a very well-known example. This is an X-linked disease, and boys suffering from this mutation, they don't have an immune system at all, and they may have to live in bubbles. And for these, also gene therapy is being developed to restore that particular gene. Allergy is an overreaction of the immune system, and it can be very broad to many different antigens. Chronic inflammation, many people suffer from chronic inflammation, pain throughout life, as an overreaction, but not heavy enough to cause death. It becomes chronic and takes years. So let me go into more detail. First of all, what are the organs of the immune system? Because it's a system, it's all over our body. So we have what we call primary immune organs, and that is the bone marrow, here exemplified by cells in the long bones, but it's also in the hip or in the sternum, and the thymus on top of the heart. These two organs are called primary organs, as these are the sites where the immune cells are being made. And immune cells are the white blood cells coming from stem cells in the bone marrow. And almost all cells, and that will be the next slide, develop in the bone marrow, except for T cells that migrate to the thymus, where they then will further develop. Inside our body, we have other parts of the immune system, and that is called the secondary lymphoid organs, the tonsils, lymph nodes throughout the body, the spleen, virus patches in the intestine, everywhere where you can encounter foreign antigens, this may happen. So these cells, as I said, there are hematopoietic stem cells in the bone marrow. This is where they reside, and depending on signals, they divide in, and then you can find many different graphs in literature, but you will get in the end all kind of lineages of white blood cells. The red cells and the platelets also come from the stem cells in the bone marrow, but do not belong to the immune system. Red blood cells, of course, carry oxygen, and platelets are very important for coagulation if you have bleeding disease or if any damage to your skin or whatsoever. So we have a series of innate immune cells. Granulocytes, neutrophils, eosinophils, and basophils. We have natural killer cells, or monocytes, and these reside usually in the blood, and macrophages, dendritic cells, in the tissues. And they're all sitting there to find the first invaders and then cause acute or later chronic inflammation. These dendritic cells can present antigen that they have taken up to T cells and B cells of the adaptive immune system. So the T lymphocytes, the T cells, we can divide in several cells: helper cells, T helper cells, regulatory T cells, which are also kind of helper cells, but regulatory helper cells, and cytotoxic effector cells. So cytotoxic T cells will kill and are helped or regulated by the other T cells to be functional. These helper T cells also can help B lymphocytes to become plasma cells that secrete antibodies. And antibodies are very important. Antibodies are very important in fighting many diseases and are also used in the cancer treatment, as I said before. The innate immune system really starts in the first few hours after a microbe has breached the epithelial barrier. Phagocytes complement and NK cells, natural killer cells here, play an important role in fighting. But as I said in the previous slide, usually this is not enough, and the dendritic cells present antigen to T and B cells in the adaptive immunity that make the effector molecules or effector cells to fight the disease. But that usually takes days. So the immune system is really divided in place and in time. So the innate immune system, and this is a similar example as I showed you previously, when it's non-activated, here you have the skin, the epidermis and the dermis, cells are waiting, type of dendritic cells, some memory T cells, and in the blood vessels beneath, the granulocytes, monocytes, and natural killer cells are floating. And upon damage, injury, when an antigen comes in, inflammation starts. These Langerhans cells, a type of dendritic cell, will become activated, start to secrete cytokines. They start to eat the pathogens coming in here, but they also start to attract already granulocytes and monocytes and natural killer cells, which is not on this picture. And they have the first response. These dendritic cells, after they have eaten the antigens, they migrate through the lymph node, activate T cells that then will go to fight off the final danger. So that are the most important functions of the innate immune system. So let's go a little bit through these effector mechanisms, like barriers, soluble factors, phagocytes, as well as the killing mechanism, the killing cells. So these barriers, well, our skin is our biggest barrier on the outside, or the intestine, or the lining on the inside of our body, from our mouth to the anus. There is also a very tight controlled epithelial barrier. And this really protects us from invaders. But once there is a breach, either on the outside or we inhaled and we get bacteria, for instance, in our gut, there are epithelial cells that can produce peptides or antibiotics that also will help killing these invaders. And we even have intraepithelial lymphocytes that look like T cells that can recognize and kill off the microbes. But you understand this is not enough, so we have soluble factors. And these are the most important. Interferons, there is the mannose binding lectin. Mannose is a very important component of bacterial cell walls, and lectins can recognize that. And later on, you have complement. So let's go through these different factors. What do they do? Well, first of all, if a cell becomes infected, this cell starts to produce type 1 interferons, interferon alpha or interferon beta, as a first danger signal. Based upon this interferon production, viruses cannot infect neighboring cells that easily. So these cells cannot spread the virus anymore. And also natural killer cells become activated by the type 1 interferons. And they start to do their function, really killing naturally. And then the infected cells will die. Because of these dying cells over here, dendritic cells will pick up remnants of these dying cells, take it to the lymph nodes, and they're presented to the helper cells. So this is all due to the initial secretion of the interferons. Now remember this until the end when I start talking about the coronavirus again. Complement. Complement is a series of 9 different proteins, and they work in a cascade. Well, first of all, there are 3 ways of activating complement. The classical pathway is called the classical pathway because it was firstly discovered, but it needs antibodies secreted by B cells that opsonize bacteria, and after that it becomes activated. The alternative pathway was only later discovered, but is actually easier. It's direct recognition of the invaders, the pathogens. And as I said, mono-binding lectins is a third way of activating complement. There are some different molecules involved in this part, but they all lead to the activation of a complement factor called C3. And an activation means that it is split into 2 molecules called C3b and C3a. And C3a is a molecule really causing inflammation, activation of many parts of the immune system. And C3b is then deposited on the microbes that opsonizes them, giving them available to macrophages for phagocytosis. And I will come to that also a little bit later. This then will activate C5, C5 gets C6, C7, C8, C9, and you get a membrane attack complex. So this is the combination where C3b activates 5, 6, 7, 8, making 9, and getting a whole complex, actually making really holes into the microbes. That causes the bacteria to lyse. And that is nicely shown in this picture, where you really see the holes in the bacteria under a large microscope, electron microscope. So this whole complex, C5 to C9, is called the membrane attack complex. Phagocytes. We know a couple of them, and they're all recognizable by their morphology. On the left-hand side, this picture, you see in the blood smear, between all the erythrocytes, a neutrophil or a neutrophilic granulocyte. You can recognize that by the multilobular nucleus and a neutral colored in the staining, a neutral colored cytoplasm. And that's why it's called a neutrophil. A basophil will be very purple, and an eosinophil will be orange, also with the multilobular nucleus. In the middle, you see a monocyte. The monocyte is usually a little bit bigger, but it can be easily recognized by this horseshoe-shaped nucleus. And on the right-hand side, this rather dimorphous cell is a megakaryocyte. The neutrophils, as you see here on top, in this picture, the multilobular nucleus, is the most abundant white blood cell. We have about between 4,000 and 10,000 of them per milliliter, and it comprises 1% of our total blood volume, but it also comprises about, the white blood cells themselves is about 10,000 or 1% of the blood volume, of which half to 75% are neutrophils. This is the most abundant and the most fast reacting cell. It has different functions. It comes from the blood, and when it's alarmed, when the danger signals have been there, microbes have invaded, it starts to upregulate molecules on its surface, whereby it really adheres to the endothelial cell wall of the blood vessel. It can extravasate and comes into the tissue. This is all based on chemotaxis to these molecules, to these danger molecules. And then they can secrete cytokines to attract other cells. They secrete cytokines to attract other cells. Or they will first do the phagocytosis, after which they produce reactive oxygen species. By the reactive oxygen species, the microbes will be killed. And when that does not work, neutrophils also can secrete these nets. Net formation will occur to trap microbes, and then other cells will be recruited. Usually neutrophils die very quickly. They have a limited lifespan. When once activated, once secreted their oxygen species or the nets, they will die. And that is also what you see, for instance, in this skin inflammation. And the yellow pus that comes out of it is basically dead neutrophils. Other important phagocytes. The monocytes come from the blood and in the tissue become macrophages. So we know, depending on the tissue, many types of macrophages. They can be in the bone marrow, in the lymph nodes, are active by the T cell cytokines, and in the liver, they're called Kupffer cells, or alveolar macrophages in the lungs, glial cells in the brain, and even in our bones, eating the bone, restructuring the bone, the osteoclasts. All these cells are macrophages. You see on these pictures they have different shapes. They have very similar functions all over. So what do they do? Well, they do many things, but they have to have all kinds of receptors to be able to do so. So first of all, they have receptors for chemokines and cytokines. Chemokine receptors will recognize chemokines secreted, and that causes their movement to the place where they are needed. Cytokines may activate them, may induce them into proliferation. Then they have a molecule of CD14 and toll-like receptors. These toll-like receptors, I will come to also in a minute, are very important receptors recognizing pathogens. These are non-specific, but still there is some specificity in there, and that will come in the next few slides. But these receptors do recognize the antigen. I already introduced complement. The complementary receptors are also present on these cells, allowing microbes to attach to them, opsonize them for phagocytosis, or also antibodies that can recognize microbes will be opsonized, these microbes, and can recognize by Fc receptors. And Fc is the tail of an antibody, and we also come to that later on during this lecture. But by this recognition, microbes can be phagocytosed. These are these receptors. We have different pattern recognition receptors. So they're not so specific, but they do recognize patterns. And we have many different types, in total about 100. The so-called extracellular toll-like receptors. From the toll-like receptors, we have around 10, and here you see examples of toll-like receptor 2 and 6, so they can form heterodimers or like TLR4 homodimers. We also have C-type lectin receptors, intracellular toll-like receptors, RIC receptors, and not-like receptors. So many different classes recognizing different patterns, but in the end they all lead to a molecule called NF-kappa B, which is a transcription factor that will go to the nucleus and then starts to transcribe to activate pro-inflammatory cytokines as well as the type interferons. Here you see again the toll-like receptors, either as hetero or homodimer, and these are the patterns that it recognizes. So there are different sets of different combinations of toll-like receptors that recognizes parts of bacteria, lipoproteins, lipopolysaccharides, the flagellin, certain bacteria have a tail, or bacterial DNA. And the big difference between the bacterial DNA is that it's not methylated, while human DNA is methylated. So that is the difference in recognition, how our body recognizes bacterial DNA versus human DNA. Or from viruses, that can be double-strand RNA, single-strand RNA, we have the intracellular toll-like receptors that all go to the NF-kappa B and activation of pro-inflammatory cytokines. And I think this is an important picture to remember, which is a very generalized picture, and it really depends on which signal binds to which receptor, what's happening next. But as a generalization, you can say that after activation of the receptor, NF-kappa B or other molecules inside the cell will become activated, phosphorylation occurs, transcription factors are either being formed or degraded so that the real transcription factor is allowed to go to the nucleus where it binds to a responsive element in the target gene that then by the RNA polymerase is transcribed into messenger RNA. The messenger RNA goes to the cytoplasm where it's translated into protein that is secreted that will change the cell function. So this is a very, very standard way of how cells react to signals from the outside. Really depending on which signal comes, the reaction changes. This is something, an important mechanism to remember. So in these phagocytes, these different receptors will do different things. As I said, when you have, here is the mouse. When chemokine comes, it binds to chemokine receptors that causes migration into tissues. Or the mannose receptor can bind directly to the mannose binding lectin binds to these microbes and you get phagocytosis that ultimately will lead to killing of the microbes. But also the toll-like receptor in combination with CD14 causes induction. Cytokines secreted in the environment will be recognized by cytokine receptors that all lead to production of oxygen species nitric oxide, killing of microbes. Cytokines are being produced by these phagocytes that will then activate T and B cells of the adaptive immune system. Or even fibroblasts can be activated that ultimately lead to tissue repair after the damage is cleared. So these are some examples of how different molecules bind to phagocytes and give different results, all according to the mechanism I explained to you in the previous slide. So what is phagocytosis and how does it work? Well, if a bacterium is getting recognized by the receptors, the membrane really engulfs the bacterium and it's being taken up in a phagosome. The macrophage already contains lysosomes in which many different molecules are present. And these combine to a phagolysosome, leading to destruction of the microbe and exocytosis. So what is happening inside this phagosome? Well, here are some mechanisms of the phagosome. By the vacuolar ATPase, hydrogen molecules can come in, or the NADPH oxidase, oxygen radicals are formed. The phagolysosome already contains lysozyme, or other different hydrolases. Here it says about 50 different types. Or also the NO synthase can make nitrogen radicals. So all these mechanisms together can produce enough damage to the microbes to die and then be secreted by exocytosis. There is a nice movie on YouTube that you can see this process in reality. So have a look at it. Natural killer cells. As their name says, they're born to kill. That was their first function that was described. And here you see a natural killer cell attacking a tumor cell. But now we know that natural killer cells have two functions. Not only the killing of virally infected cells or tumor cells, but it's also very important in secreting cytokines. So once a macrophage has a phagocyte, a microbe, it starts to produce IL-12, which activate natural killer cells to produce interferon gamma. And the interferon gamma is then a positive feedback loop to the macrophage to help assisting in the killing of that engulfed microbe. I will talk now about the killing function. So why do they not kill like they're in the body if they have the name of natural killing cells? Well, they are kept at bay. A normal cell is protected against killing because natural killer cells also have inhibitory receptors. So in general, a normal cell have MHC class 1 molecules. This is another molecule that is very important and I will explain later during this lecture. But in this case, it's important to remember that it is a signal that activates the inhibitory receptor so that the NK cell will not kill the normal cell. There are also stimulatory ligands that can bind to stimulatory receptors, but the balance in a normal situation is that the inhibitory signal is stronger than the stimulatory signal. But it may that an infection or a transformation may occur. In that case, the inhibitory signal is not there anymore. MHC molecules may be downregulated. In the case of tumors, this has been described very well. Or the activation signal may be upregulated, and thereby changing the balance between inhibition and activation in favor of activation. So then the NK cell can kill the virus-infected cell, or it can kill a tumor cell. And this killing occurs by granules that these natural killer cells have. These granules contain perforins that will make holes inside the target cell. They perforate the target cell and then secrete other type of molecules called granzymes. And granzymes, they go through the pores into the target cell where they activate a cascade of caspases, ultimately leading to apoptosis and death of the target cell. There are some other molecules called FAS or trail that also give a death signal by activating cascades. This is perforin granzyme independent and may take a little bit longer to become activated. So once the granzymes have finished their work, FAS and trail may take over in ultimately killing the target cell. Here you see a little bit better what is inside the cell. The granzymes comes, they activate this cascade leading to DNA fragmentation. And the DNA fragmentation, of course, is detrimental for the cell, and the cell will die. So let's move on from the innate to the adaptive immune system. And the big cell to do so is the dendritic cell. Here you see various pictures various microscopes of techniques of how dendritic cells look like. They have a good body, but many of these dendrites. And with the dendrites, they feel, they sense, and they also can present. Also, dendritic cells have these similar pattern recognition receptors, like the macrophages I described before. So also dendritic cells will either on the outside or on the inside recognize danger, activate the NF-kappa B, leading to pro-inflammatory cytokines and type 1 interferon gamma, interferon alpha and beta. It really depends on the signal, what kind of cytokines will be produced and what kind of response will occur. So the dendritic cells are located at places in our body where they can encounter antigen. Anywhere in the skin, in the mucosa, in our body, in many, many places. So there they sense with their dendrites and they find the microbe. Start to produce inflammatory cytokines. They will move through the lymph node, through the lymph vessels to lymph nodes where they have the antigen that they picked up in the skin present to T cells. And the T cells start to proliferate to be able to do their function and move back to the site of infection. To do so, and to discriminate between self and non-self, a dendritic cell needs to activate a T cell by 3 signals. Signal 1, signal 2, and signal 3. So first of all, let's start how this comes about. There are different types of pattern recognition receptors. So depending on the pattern, depending on the microbe that's being recognized, you get different type of signals. So the microbe is taken up, is being broken down, and is presented in the MHC class 1 or class 2 molecules to a T cell receptor. So this little green dot, that is the peptide of the pathogen. And here is a very specific interaction. And I also will come to that later, how this works in more detail. But this gives signal 1. This is specificity of the reaction. Then depending on the pattern, different types of molecules are being upregulated. First of all, co-stimulatory molecules. CD80, CD86. Co-stimulatory molecules bind to CD28, delivering signal 2. CD40, CD40 ligand is upregulated, and many cytokines are being produced, either Th1 or Th2 polarizing cytokines. That really depends on this naive T cell to move into a Th1 cell or a Th2 cell. Th1 cells secrete different cytokines compared to Th2 cells. And Th1 cells mainly activate the cytotoxic T lymphocytes. And Th2 cells mainly activate the B lymphocytes. So these 3 signals are very important. This is a picture I really would like you to remember and to think about how that works, because this is instrumental. And also here you can find on YouTube a nice movie. After recognition by the T cell receptor, you get this whole cascade leading to factors transcribing to binding to the DNA, resulting in transcription of mRNA, as I said before. And these are molecules you don't have to remember, but at least try to understand that after the signal comes, this is what's happening inside the cell. But it results in what we call cell-mediated immunity. The T helper cells, they will help to phagocytose. They will help the cytotoxic T lymphocyte to become a very good killer. They also may help B lymphocytes to make antibodies. And the B lymphocytes making antibody is what we call humoral immunity. And if we have enough antibodies, that also will help to block infections. It binds to the microbes and it prevents recognition and entry into the cells. So how do the receptors on lymphocytes, on the B and the T lymphocyte, look like? On the left-hand side, we see the B cell receptors and the T cell receptors. And every T and B cell have a unique antigen receptor. They are generated by gene recombinations. And this recombination is independent of the antigen. So a B cell receptor, or an antibody, is comprised of two, this is one, and in red is the second heavy chain, as well as here in green, two light chains, making a structure of a protein of around 150 kilodalton with one and two antigen binding sites. This is important so that cross-linking can occur. One antibody can bind on different parts of two different antigens of the same molecule, of course, because the specificity is enormous. The upper part is what we call the FAP region, and this is called the FC region. I mentioned FC region before because this is the part that has to bind to the FC receptor on the granulocytes or on the macrophages, by which the opsonization occurs. So recognition of the pathogen, FC binding to FC positive cells, and uptake of the microbe into the phagocytes. Or it may be membrane bound, not secreted but membrane bound. That's especially in the beginning. The T cell receptor is expressed on T cells. It looks a bit similar. It has two chains, an alpha chain and a beta chain, but it has only one antigen recognition site. And this is the peptide presented in the MHC. Making this recombination to get the unique antigens receptor is a very complex process. It has been discovered in the late 80s, and a Nobel Prize has been awarded for this mechanism. Because if we, for every receptor, would have a single coding gene, our DNA would be too large and our cells would be too small to have all the DNA. So we have a genomic organization of both the heavy and the light chains, or the T cell receptor genes, in a very similar way, where we have so-called variable regions, diversity regions, joining regions, and constant regions. When we go back to the previous slide, you will see that the constant regions code for this area, whereas the VDJ code for about this area. When we have a particular B cell, this is the example for the heavy chain of a B cell receptor, but it works exactly the same for all the other chains. So we have the organization of V1 till Vn. It depends a little bit on the chain how many of these segments we have. The same is for D, J, or C. So we get a somatic recombination on the DNA level, in the genomic DNA. In the first recombination event, in this particular B cell, D1 and J1 recombine. In the second step, D1-J1 recombination occurs to V1, and the intermediate DNA, which is exampled here, is just being spliced out and kicked out of the cell. Then there is a genomic recombined piece of DNA that's being transcribed in RNA, followed by a splicing event of all the different remaining regions. so that the VDJ recombined DNA piece at the RNA level comes close to the constant coding domain, CMU. And this creates a unique B cell receptor. And in this other B cell line, randomly other molecules get recombined together. And this also happens for the light chains and for the alpha beta receptors. And as we have many different numbers of these V, D, or J segments, there is an almost endless repertoire being able to be generated. And this is what 10 to the 11th or 10 to the 16th possible B cell receptors or T cell receptors can be generated. And that theoretically then will lead to possible T cell and B cell receptors that recognize self, that recognize own antigens, own molecules in our body. And if they are not eliminated, we have autoimmunity. So we need to have a system that learns our body to recognize self versus non-self. And for T cells, that is the thymus. This is the microenvironment where T cells develop. The thymus is located just above the heart, and especially in young kids, it's very large and it shrinks when we get older. This is a picture of a thymus and just stained by a blue dye. You see a dark area which is called the cortex and a light area which is called the medulla. And that is then in this drawing also pictured. So in the medulla, in the cortex, you have cortical epithelial cells in light blue and many young thymocytes. There is a sparse macrophage, and in the medulla we have medullary epithelial cells, less thymocytes, and we also have more macrophages and dendritic cells. And they have functions. This is a nice picture of a thymus where you see the three-dimensional epithelium with small thymocytes crawling through it. So this is very unique to the thymus, where epithelium is three-dimensional. This gives them really a large surface that they can scan for interaction. What is going to happen? Well, first this summary slide. Cells, young progenitor cells, come from the bone marrow through the blood, and they migrate into the thymus tissue at the border of the cortex and the medulla. Here they are called DN1, and DN means double negative. And that's double negative for CD4 and CD8. Based on chemotaxis, they migrate to the capsular area, maturing according to stages of double negative 2, double negative 3, recognized by other molecules, and they really start to proliferate. During this proliferation phase, they also start to rearrange first the beta chain and later their alpha chain. They start to upregulate CD4 and CD8, becoming double positive for CD4 and CD8. They lose the capacity to become gamma delta cells, NK cells, NKT cells, dendritic cells, and at this stage they're really committed to become T cells. And then they have to go that selection process. Are these alpha and beta chains that have been rearranged good or not good? So first there is a process of positive selection on cortical epithelial cells. When they have a good T cell receptor, they're allowed to move to the medulla, becoming single positive, downregulating either CD4 or CD8, and they will undergo a process called negative selection. Do they recognize self? And if they do, they are negatively selected. And only the ones that remain are allowed to move out of the thymus and become in the periphery. This is about 5% of the cells that undergo a successful process. So now into more detail. What's happening in this cortex and medulla in that selection, the positive and negative selection? Well, positive selection is that randomly generated T-cell receptor recognizes MHC class 1 or class 2. It's functional, it's allowed. A negative selection is that the generated T-cell receptor recognizes a peptide, that you see here, peptide in the MHC. So here you look from above to the MHC molecule, you see a peptide present in the MHC. When that's being recognized as a self molecule, the cell will go negatively selected. So again, there is a random expression of an alpha-beta receptor on the precursor that is CD4, CD8 negative. It upregulates during the proliferation phase and alpha-beta chain rearrangement the CD4 and CD8 molecules. So at this stage, the cell is double positive and has a low affinity alpha-beta receptor. It starts to scan the cortical epithelium. Several things may happen. When it's a double positive cell and it recognizes MHC class 1, that's great. Cells will downregulate CD4 and the cell becomes a CD8 single positive thymocyte. Also, this double positive cell or another double positive cell may recognize MHC class 2. The opposite happens. CD8 is downregulated and the cell becomes a CD4 single positive cell. And if in a certain time, which is usually about 2 weeks, there is no engagement with the MHC, there is no recognition of the T-cell receptor to the MHC, the cell goes into apoptosis. And here you really see that close interaction of a pink thymocyte to the purple epithelium. So now, now these single positive cells have come to the medulla. The affinity has increased of both MHC molecules. So several things again may happen in again 2 to 3 weeks. So the CD8 positive cell is looking for peptide in MHC. And when it's after 3 weeks has found no interaction with peptide in class 1, it is allowed to stay. This will be a nice CTL that is allowed to go into the periphery. The same is true for CD4 positive cells. When they do not find self-peptide in the MHC, they are allowed to mature and go into the periphery. However, when these cells recognize self-peptide in the MHC, it may be helped by dendritic cells and macrophages. We don't want these cells. They recognize self and they're dangerous. So these cells should go into apoptosis. "5% of the cells leave the thymus and the others die." So let's now talk about in close detail about this MHC. Here is the peptide presented in the MHC to the T-cell receptor. And here you also may appreciate and understand the specificity. This is really a three-dimensional structure where amino acids, either here the crosses, the balls, or the triangles, fit into the groove of the MHC. And when this is not fitting properly, it will fit in another MHC, or not at all. And part of the peptide is also presented to the T-cell receptor. So here you understand the enormous specificity of these receptors. in comparison to the pattern recognition receptors on the innate immune cells. So there is an even larger heterogeneity on the MHC. And the MHC being the molecule to present the peptides in signal 1 next to the signal 2 and 3. So MHC, major histocompatibility complex. MHC is a general term. In human it's called HLA, human leukocyte antigens. It's located on chromosome 6 and it has many molecules involved. Here we have class 1, class 2, and we even have class 3. Class 1, usually the HLA A, B, and C, or the class 2, here A, B, and C, or the class 2 molecules HLA, DR, DP, and DQ. You can find on this website, but I already did that for you, how these molecules look like and how many we have. But first, the general structure. MHC class 1 is comprised of an alpha chain, 3 domains, stabilized by the beta-2 microglobulin. And it creates, by the alpha 1 and alpha 2 domains, the binding groove where the peptide is being presented. Class 2 molecules are a bit different. They are comprised of an alpha and a beta chain, whereby the peptide binding groove consists of the alpha 1 and the beta 1 domain. But structurally, these two molecules look very similar. The peptide is presented in the groove to the T-cell receptor. And also these molecules, they belong to the same family as the T-cell receptor and the immunoglobulin receptor. What is so special about the MHC? Well, the MHC is the only molecule in our body of which both alleles make a protein. So both the allele you inherit from your father as well as from your mother makes a protein. And the importance is that it increases the number of different MHC molecules that can present peptides to T cells. HLA or MHC is also the most polymorphic gene in our body. And it means that we have many different alleles present in our population. So our population as human, we can present almost all peptides to T-cell receptors. But as an individual, we respond differently. And this also may explain why certain people will die of a virus infection and others do not. But this system ensures that we as a human species will survive. So here is really the difference between the human as a species and the human as an individual. And how many do we have? Well, we really have many. This is from the website I earlier indicated to you. In the past 3 years, on the DNA level, we have seen an increased complexity from class 1 and class 2 alleles. And here you see the different numbers. And this is a rather recent one. In the first part of this year, we now have about 19,000 different class 1 alleles and over 7,000 different class 2 alleles. And these are really differences on 1 base or 2 base differences. On that high resolution we can determine. But 1 base, depending on where it is in the triplet of coding the amino acid, may change the conformation of the HLA molecule. It may change the affinity of the peptide that is in that groove to present. Same is true for the class 2 alleles. Also here in 2020 we have many different class 2 alleles. Not as many as class 1, but also a lot. creating an enormous diversity of peptides we can present. And where is this polymorphism in the MHC molecule? Well, you see, it's not randomly distributed. It is at certain hotspots. And these hotspots actually are mainly in this groove. Because here we look again on the top, or here on the side, but most of the mutations are in these, or the polymorphism, I must say, are in the groove where they present the peptide. The last important thing about mentioning about the MHC is on which cells is it expressed. Well, class 2 molecules are only expressed on antigen-presenting cells, dendritic cells, macrophages, and certain type of B cells. Whereas class 1 molecules are expressed on all nucleated cells. So the CD8 positive T cell can infect any cell, can kill any cell that has been infected by a virus. So it's important to realize this. The MHC, this is the standard pathways. Extracellular molecules, microbes are being endocytosed by a phagocyte. Lysed peptides are being loaded in the MHC and presented to the CD4 T cell. Because remember, phagocytes are the ones that can present antigen to T cells, the dendritic cells, the macrophages. When a cell gets infected by a virus, the virus also inside the cell gets processed, and peptides are loaded in the MHC, and it's presented mainly to the CD8 positive T cells. So these are the classical pathways. But in recent years, we learned that also extracellular microbes can go into the class 1 pathway, a process called cross-presentation. And fortunately, that's there. That makes us make use of it in developing vaccines. Let's go to B cells. B cells also get activated. I already said complement may opsonize a microbe, and then it gets recognized by both the immunoglobulin receptor as well as the complement receptor, and that leads to B cell activation. And of course, B cell activation is again a very complex system of all kinds of molecules reacting to each other, leading to transcription factor and functions. And these functions can again be different. After the response, the B cell has recognized the antigen, it will start to expand, will react to cytokines influenced by the helper cells, and starts to secrete immunoglobulins when they become a mature plasma cell. It always starts with IgM, and in secondary responses becomes IgG. So here we are at the adaptive immune system. What are the main effector mechanisms? Well, B cells, naive B cells, after they have recognized antigens, they will proliferate and make antibodies. That is their main function. Fortunately, some of them become memory B cells, and I will soon explain what this is. Also, naive T cells, after an antigen-presenting cell has presented peptides in the MHC, they start to proliferate and become effector T cells, and some of them become memory T cells. When the danger is cleared, the cells can go into apoptosis and homeostasis is restored. Memory. How is memory induced? Well, let's first look at the purple reaction. Naive B cells, in this example, they meet an antigen. Naive B cells start to grow, start to proliferate, start to secrete antibodies. The antibodies clear the microbe. Some of the B cells will go into memory cells, and the others will die. Then a couple of weeks, in this example later, we get exposed to two new antigens, one new and one the previous one. So we have memory in naive B cells. Well, the naive B cell that now recognizes antigen Y has the same primary response against Y. Slow response, slow proliferation, getting activation of B cells, secreting antibodies, and restoration of the homeostasis. But the cell that recognizes the same antigen, antigen X, will see a very quick, sharp rise of proliferating B cells. In a short time, we will have many B cells secreting lots of antibodies. So this response starts early, becomes very high, and lasts longer. So the memory B cell is much quicker activated. Same is true for T cells. So this is the specificity leading to memory. And memory is an integral part of vaccination. For sure, we cannot vaccinate with live viruses. I assume most of you have been vaccinated against rubella, against measles, many of these viral diseases. Traveler vaccines you may have heard of, rabies, or in Tomsk, I think in summertime you will have the Japanese encephalitis virus vaccine that may be useful. But anyway, there are many different types of vaccines that can be made depending on the way of inducing an immune response or on the original pathogen. That's exemplified here, the different type of vaccines we have against the different, mostly viruses, but sometimes bacteria. And by giving these attenuated vaccines, killed vaccines or whatsoever, we usually induce this immune response. We do not get sick, but once the real pathogen comes, we are protected against. And that brings me to the coronavirus. As you may have seen in this list, there is no vaccine against coronavirus. So now we are being exposed in this pandemic all over the world to the coronavirus. It becomes important. So what are coronaviruses? Well, they're enveloped, non-segmented, and they have a single-stranded RNA genome. We know about 30 different coronaviruses in 4 genera, and the alpha and beta subtypes can infect humans. And we have about 4 or 5 viruses coronavirus that will infect us, and they cause simple colds. They've been around for a long time. We do get colds once in a while, and some of them may have been instigated by a coronavirus. In general, all coronaviruses bind to receptors. And the current virus, the SARS coronavirus 2, but also the SARS coronavirus 1 that caused SARS about 17 years ago, bind to a receptor called ACE2. And ACE2 is expressed on alveolar cells, and that will come in some next pictures as well. So these cells fuse to the membrane, the RNA genome is being replicated, new virus proteins are being made, translated into the proteins, and you get a new virus particle. And you get many virus particles, and that is being released. So this is usually how coronaviruses replicate. Some clinical symptoms. If we go on top, this is the healthy situation. Here you have type 1 and type 2 alveolar cells. The type 1 cells allow for oxygen and carbon dioxide exchange. The type 2 cells are cells that secrete mucus and protect and the surfactant, keeping these bowl-shaped structures intact. But the virus, once we breathe it in, they go down to the alveoli, where the ACE2 receptor is highly expressed on the type 2 cells. So the virus attaches to the type 2 cells and start to replicate, and thereby also destroying these cells. As a result, interstitial fluid will go into the alveoli, and that causes the symptoms of difficult to breathe. They fill with water, and that's not in this picture. They also seem to fill with blood, because bradykinin is also remaining in these alveoli. It's not being captured by the ACE2 receptor. Usually bradykinin, an inflammatory response, binds to ACE2, but ACE2 now is gone because it has taken up the virus. So also bradykinin remains in this fluid, and blood comes in, causing even more severity to breathe, and people go to the intensive care units. If the virus has infected through the ACE2, what type of immune response will come? What do we know by now? What is fighting the disease? Well, first of all, and remember I said that in the beginning, the initial response is that type 1 interferons are being secreted by cells infected with viruses. Well, it seems that in certain people, this interferon response is delayed or suppressed, thereby not activating neutrophils, not activating natural killer cells, or macrophages, very low. And that may cause the severity of the disease. But once activated, of course, these macrophages and neutrophils become active and try to kill the virus. Macrophages, dendritic cells, will activate B cells to become plasma cells that will secrete antibodies that also will protect viruses from binding to the ACE2 receptor. This is a bit more detail what the immune response is about. The COVID virus will bind to these receptors. Pattern recognition will activate pattern recognition receptor. And this is the spike protein, the outside on the coronavirus binding to TLR4. So you see there's several ways on a molecular level that receptors get activated. You get signaling through MyD88 or TREF. The interferon responsive elements will take care that the pro-inflammatory cytokines or the type 1 interferons are secreted. NF-kappa B is being formed, which will lead to activation of secretion of interferon gamma that will activate T cells and natural killer cells. That are an important part of the immune response against the virus. Neutrophils are very important cells against clearing the virus, and the B cells making the antibodies. So there is again a whole cascade that goes into play to fight the coronavirus. But as the virus is clever, as I said, it sometimes may block the interferon alpha production, leading to a lower expression. It also may block T cell functions or neutrophil functions, all leading to the severity of the symptoms. I see a question from Evgenia. What does the recombinant vaccine mean? So I go back to this slide. Recombinant vaccines is that we don't use whole viruses anymore. They may be dangerous, like in the HIV or Ebola or also the hepatitis viruses. They're way too dangerous to just use live attenuated or killed inactive vaccines. If just one virus particle remains, it may cause the disease. So once we know, and that is maybe then a good example to show on the coronavirus, here is that spike. If we now only use that spike DNA and put it in a harmless recombinant molecule and make an artificial virus, then we can induce an immune response against the spike without having in that particle the nucleic acid, the information of the virus. It cannot replicate, it cannot do any harm. So what is next against this coronavirus? At the moment, we don't have a cure and we don't have a vaccine. So yeah, social distancing is what we do. And that's what in the Netherlands they call the new normal. But nobody likes this. We are releasing these social distancing a little bit, but we are very scared, and I think all over the world we are very scared that there will become a second outbreak until we have a vaccine or a drug. So it's very important, and many institutes and companies are now developing drugs and vaccines. And having said that, we have discussed, and we would like to give you an assignment. What kind of vaccine against coronavirus would you develop? And I think with the assignment, it's good to work in two groups. So divide the student group in two. And please make a project where you describe the rationale. What type, I introduced you a couple of vaccines. What kind of vaccine would you develop for the corona SARS virus 2? Please describe why you choose this, what kind of background based on what you would like to develop your vaccine, and what is the immunology behind it? How does it work? But next to that, I think on the world level, but also on the practical level, is a very important question. How will it be produced? How can you make a vaccine? How can you make vaccines in such quantities that now the whole world can make use of it, but still having the quality? How do you prove for a vaccine that it works, that it has the quality, and it has the safety? Because you must imagine that for many diseases, like cancer, we can use drugs, but many cancer patients will die anyway. These people are sick. So there are different safety issues regarding. Now with a vaccine, you are treating healthy people. And one death of a healthy person is one too many. And that's a different state of mind than when you're treating very severe patients. Then it's an incalculated risk. So please describe these things. And we would like to ask you to make a PowerPoint presentation of it that will last about 15 minutes. And please send it to email, either to Evgenia or to me, and to present it at the next webinar. We still have to define the time point, but you will get that from the course coordinator when that will be. And then we have a discussion on the vaccine that we have. And I would like to encourage you to ask questions from one group to the other, and of course answer the questions. And I also will try to discuss it with you. Maybe we come up with some clever ideas. Find it in the literature, find it out. There is enough. I've been searching. I know there is a lot of work being done, and possibilities are being searched and investigated by people. So I hope that the next time we can come up with some nice idea. Basic literature, I always recommend these two books, written by, on the left-hand side, by Abul Abbas. It is a really good textbook showing all kinds of mechanisms, whereas on the right-hand side, written by Reut, it also shows the animal experiments that lay beneath all the theories. So with this, I would like to thank you for being here. If you have any questions, I think we can go over to the question and answer session. And here also please find my email address if there is anything you'd like to know on what I said. And I think I give the word back to Max, who will be our discussion leader.
Yes, dear Professor Germeraad, thank you very much for an interesting lecture. And yeah, now we have some time for the question session. And I already see one question again from Evgenia Solovina. So, Zhenya, maybe I can give you the word and you can switch on your microphone and just ask your question.
Yeah, Wilfred, thank you very much. I'm so happy to see you again. Can you hear me?
I hear you very well.
So yes, my question was about flu vaccine, because some people say that these vaccines really help, but some people say that they do not work, and since the virus can mutate, there is no need to have another vaccine every year. So what do you think? How could you explain this phenomenon?
That's a very nice question. Flu is a special case. It's a bit longer story. The influenza virus mutates a lot. It also has core proteins called H and N. So you may hear about the subtypes H5N1, which was the flu bird. And having said flu influenza virus or bird flu, that is another part of mutations. So there are two types of mutations with the influenza viruses going on. And that hampers making vaccines. So there is an antigenic shift and an antigenic drift. The antigenic shift is an easy one, that the individual viruses mutates, but the antigenic drift is that two different viruses come together in the same person and mix. So this is often said in Hong Kong or China, where this often happens, where the markets, also like the current coronavirus, animals and humans live so close together that in a single animal, a human virus and a bird virus come together and the viruses mix and create a new virus that the human population hasn't seen before. So this is what we never know when that happens, but we do know that the mutations in one virus will happen basically every year. And that is because these viruses have a lack of DNA and RNA repair mechanisms. So once a virus replicates its DNA, it's not being checked whether it's correct or not. So you get new different H molecules, you get different N molecules. And that means that in the end, the surface of these molecules, they change. And the antibodies formed in the year before do now not recognize this molecule anymore. So what we try to do with the influenza vaccines is to predict which will be the virus strains next year. And that has to do with the number of viruses per person in the population. That we try to predict, well, this will be the H type or the N type that will be around next year. So then a vaccine is being made based on three of these molecules. And when the viruses behave like we predict they will behave, we are, after the vaccination, protected against these viruses. But as you know, who can predict the future? I think nobody. And the viruses surely don't listen to what we tell them to do. They may mutate in a different way. And when they mutate in a slightly different way, we might be partially protected after being vaccinated. You do get a B cell that start to make antibodies that will bind and prevent infection. But when the mutation becomes too big, there is no B cell, there is no antibody that recognizes this virus strain, and we get sick. Let alone when that bird flu has recombined with the human virus. And then we have no protection whatsoever. That was the scare we had a couple of years ago with that H5N1 type of virus. It has never been in the human population. So nobody of us had protection against H5N1, which is the same situation with the coronavirus SARS-2. Nobody has seen this virus, so nobody has protection. And that's the whole scare we have, is that we all may die. And that's why if you isolate the people and have them apart and can control the infection rate until some people do, and now we know after 2 months people have recovered, they may have built up immunity, B cells may have responded, made it antibodies, and these people now probably, we still don't know, that will be solved in the next couple of weeks, months, may be protected against the second infection.
Thank you very much. Yes, thank you very much. Maybe students can also ask something.
So, dear students, do you have any questions to Professor Germeraad? So I would say that there are only two options. Everything is clear and nothing is clear.
Yeah, usually, or they're too shy to ask. That's the third possibility.
Yeah, maybe then I can ask you one small tricky question. Maybe you have also heard that recently in the news that it was said like this coronavirus was completely artificially produced in one of the labs, either in China or in USA. And how do you think, is it possible for us, for the humanity, to produce such type of virus from the scratch in some research lab?
Yeah, well, first of all, let me say that I don't believe that it is man-made. These are all the stories that go about, all kind of theories. Sure. On the other hand, I think it is possible, technically, because well, these viruses are not so big, and we can synthesize DNA the way we want it. So we can make, again, I think theoretically, any type of virus. And if you do that, and you play around with the genomic sequence of the coronavirus in a lab, well protected, you infect cells and see what's happening. As an experiment, you are able to do so. And then if an accident happens, these viruses may get out of the lab and infect whatever. And you can make all kind of bombs and put them in a stadium and whatever. And then I think, well, we have read, of course, many books and movies have been about these viral outbreaks and conspiracy theories that mankind can do this. So yes, theoretically, I think it's possible. What I read is that most likely, that most investigations I heard that it really started in bats, and bats somehow have an immune system that they are not hampered by these viruses. They are not killed by these viruses. They have somehow a resistance mechanism, and that's why in bats many of these viruses can live. And then there was another porcine animal that the Chinese seem to be happy to eat that got bitten by one of these bats, and then it transferred, and when the human ate it, yeah, then it became, of course, infectious to the human. That's the most likely scenario of what happened.
Okay, thank you for your opinion.
You're welcome. Well, I can understand that if people are hesitant to ask questions, well, we have been talking the last 1.5 hours, and I've been touching upon many different things as a basal overview of the immune system. And while studying on this, questions may pop. So if they come, maybe you can collect them and send them to me by the email, and I can provide some answers next time.
That sounds like a nice idea.
See if Jenny is still typing. Yeah, the immune system is one of the most difficult topics in biology. That's what we hear more often, but it also is what fascinates me about studying it and using it. But it may be the most difficult one. I understand because, as I said in the beginning, it's complicated as we have many cells, many proteins acting on different locations, different in time. It's all mixed, or it's a fluid system floating through our body, reacting where it's needed. It's not a heart that's sitting in one place and is beating. In that sense, the immune system looks much more complicated than the heart or the lungs that sit here and they expand and exchange oxygen and carbon dioxide. The fluidity of cells and proteins moving in time and place makes it so difficult. My current projects on the science is we are developing immunotherapy for cancer. So the two easiest ones to explain are dendritic cells, the ones that present antigens to the immune system. Is one cell we are using, but they're not feeding it virus proteins, but feeding it tumor proteins, in the hope that after you inject these cells into patients, that their immune response, that they really activate T cells, that the T cells will go to the CTLs, the cytotoxic T lymphocytes, will go to the cancer and eat and kill the cancer. I explained to you the killing mechanism of the natural killer cells with the perforins and granzymes. Cytotoxic T lymphocytes kill in exactly the same way, but they have to be properly activated to do so. So if we can educate dendritic cells in such a way and stimulate them in such a way that they will really activate these killer T cells, and then we hope to get better cancer treatment. We also have a startup company to do so, because these cells have to produce under very special circumstances that it is safe, and that's also why my question about safety for the vaccine, but also for the cell therapy, safety is a very important issue. So we have a new facility where we can grow these cells under very strict guidelines and cleaners, safe, so that it is safe to give them to patients. The first group of patients we will do this clinical trial early next year is lung cancer, because for lung cancer we know, well, I think two things. One is that there is hardly any treatment, and many patients still die, so there is this urgent need. And the other thing is that lung cancer is often caused by carcinogens. Smoking is of course a good example. Many of the ingredients in tobacco smoke causes a lot of mutations in genes, and actually in this case it is a good thing, because there are many mutations in lung tissue. That also means that there is a high chance of reactivity of T cells against these mutated genes, because they become more and more different than than the normal lung tissue. So there is a bigger chance that immune therapy will work. And actually that is what we already know. Patients that get certain drugs, the ones that respond better have more mutations. So that is why we are starting on lung cancer, giving tumor antigens to the dendritic cells that then will activate T cells. And another project we are working on is on natural killer cells. We do not have, we know from stem cell transplantations by leukemia or multiple myeloma, the blood cancers, bone marrow cancers, that natural killer cells can kill many of these tumor cells. So, but we do not have enough NK cells in our blood. So we have a system developed that can expand in 2 weeks NK cells, that we will have enough NK cells, and then to also give back to the patients. So that is our second project we are working on. But for some other reasons that has not scientific reasons, I must say, that has caused a delay. We do not have access to one of the ingredients anymore to do this expansion, because our collaborator was taken over by another company, and then you get in all kind of legal stuff. So we cannot expand the NK cells that well anymore, and we have to find a new alternative. If a vaccine against corona is created, shall it be tested in vivo in animals? I think that's a very relevant question, but I would like to not answer it yet, as I think it's a very good part of the assignment, especially regarding also the safety question. So we can discuss that the next time when we have the presentations. Are there any more questions? So maybe, Jenny, you can propose a couple of dates in 2 or 3 weeks from now that we have that presentation session.
Yes, I think maybe on 28th or 29th of May.
We can discuss that later, the two of us.
Yeah, okay then.
Because I have to look in my.
Yeah, I will send you the dates we will choose, and then I will also post it for students in the schedule.
Yeah. Very good. Very good. So what else? Any more questions?
So as far as I can see, there are no more questions.
Well, then I would like to thank everyone for being here and listening and staying and discussing.
First of all, we would like to thank you for joining and giving a lecture for us and for your time, especially.
You're welcome. Well, it was easy to do as I have to travel only the stairs in my house and not take the airplane to Moscow. But I would love to come back to Tomsk one day, anyway, for sure.
We're looking forward to see you here in Tomsk.
Okay, having said that, Max, thanks for hosting. And I'm looking forward to the next time.
Yep. Yes, sure. So, dear students, thank you for attending. Thank you for the lecture. And I hope we will meet in 2-3 weeks to discuss the assignment. It's also very, very interesting to me.
Very good. See you then.
So,
goodbye, everyone.